so now let us continue our discussion with respect to binding of anions. And what we observed in the last few lectures was that the factors responsible for anion binding, and also the fact that we discussed about different kind of examples. We also talked about guanidinium based receptors which actually capture phosphate anions very effectively, and we also looked at how we can modify the host so that it can efficiently bind with the anions, that is the guest, and induce high magnitudes of affinity in the different kind of systems. Now, so it's very important that when you have anion binding, electrostatics we realize plays a very important role, and it is preferable to have the host molecule which is going to capture the anions, that is the guest, to have a good electrostatic complementarity, and the host should be preferably highly charged species. And we also realized that pH plays a very important role. But in addition to that of cationic host, we can also have neutral molecules which have the necessary hydrogen bond donors, which can also interact with the anions and bind anions very efficiently. And we also looked towards the end of the last lecture, we looked at the hydrate sponge, which is essentially for a compound which has a boron center, which can sequester the hydrate ions, which is equivalent to that of a proton sponge, where we had the NMe2 group, which with the lone pair of electrons from both sides, which can capture the proton as well. So we looked at different kind of systems which function as binding sites for the anions. Now let us take up some more examples just to complete the discussion. So we saw that we have got different types of hosts, okay, which are going to bind with the guests. So to start with, we can look at charged host. So and here we also looked at organic cations. We looked at ammonium plus, and we can also have anion as gas. These are cations, organic cations as gas. So we can also have a charged species which can actually function as the host. So we can have species which have, to start with, the ammonium cation, then we can have this species as the charged one. We can have the protonated pyridine, and we also looked at guanidinium based species as well. We can now have neutral host. So we see that all the species mentioned here, they can function as a good host for binding with anions, and if they are present separately, these are organic cations which can also bind with the neutral host. So we can also have these separately as cations, they can bind with neutral hosts. Now we can also look at other neutral hosts which can bind with anions. For example, we can look at this kind of system where X is equal to oxygen, sulfur, selenium, and this can hydrogen bond with a chloride ion or any halide. We can have the pyrrole, a pyrrole based system, which also has a donor and can interact with a halide. We can have indoles, which can again bind with a chloride, and we can have urea based systems where you can have oxygen, we can have sulfur, or we can have selenium, and then we can have the hydrogen which can form NHCl minus hydrogen bonds. So, this functions as a bifurcated acceptor, and we can extend these further to squaremides. For example, we can have these kind of systems. And these can function as donors with the X minus. You can have also have oxygen or sulfur here. So, we can have this squaramide, and we can also have the thio squaramide. We can have simple amines also, and we can have semi-carbosides. So, we can have this as a donor, this as a donor, and this as a donor. So, this is a semi-carboside if this is oxygen, but if this is sulfur, then this is called thiosemi-carboside. So, you can see there are different, there is a lot of diversity when it comes to the different functional groups which can now sense anions. We can also have carbozoles. So, carbozoles are also there, and these have got a donor which can now interact with your halide. And we also have the triazoles. Okay, and we see that this is a acidic donor. We can also use boric acid, which has got these donors, and we can use activated fluorinated systems. For example, we can use so these hydrogens are all in the top, and we have all the fluorines. So, we have the hexafluoro 1, 2, 3, 4, 5, 6. So, this is the all cis hexafluoro cyclohexane. Now, you see these are the highly acidic hydrogens because of the presence of the strong electron withdrawing minus I effect of fluorine. Now, these hydrogens become acidic, and therefore, they can function as good hosts for binding of anions. And we can further activate we can further activate the substrate further, where we put a metal which can metal activated hydrogen bonds. For example, we can take a system where we have a metal. There is a metal now, and we have NH here. Now, because the presence of the metal which withdraws the electron density from the nitrogen, which in turn withdraws the electron density from this nitrogen, and therefore, that enhances the acidity of this NH. So, the presence of this delocalization, you see this delocalization is there. So, that increases the electron density on this nitrogen, which in turn pushes more electron density on the metal in a particular oxidation state, and thereby enhancing the acidity of these hydrogens, which can now function as donor atoms, and they can interact with different kind of anions. Similarly, we can have metal 3 aminopyridine based systems. For example, let us consider the pyridine, the nitrogen lone pair donated to the metal. We have the hydrogen here, and then we have the 3 aminopyridine. So, because of this donation of electrons to the metal center, the CH bonds get acidic, and therefore, they can now participate in anion binding. And we also looked at some of the Lewis acids. For example, we looked at the boron center. So, the boron center, this is now electron deficient. So, it can bind with the anions, and this is has got a lot of applications in the field of sensing of different kind of anions. For example, water can have different kind of anions, and boron based reagents can be used as sensors for sensing these kind of anions. Mercury, a divalent species. We can also have tin, which that is organo tin. We can have organo mercury. And to conclude this overall discussion about the different kind of substrates which we have, we can also utilize halogen bonding. We can also utilize halogen bonding to bring about efficient anion capture. For example, we can consider so if you have electron withdrawing groups, then that will enhance the sigma hole, and this can now bind with a halide or negatively charged species. This can bind with a halide, a negatively charged species. So, we can have species, for example, you can have diiodotetrafluorobenzene, we can have this. We can also have, because this is the sp carbon, so that enhances the electrophilicity or the sigma hole on this iodine, which can now interact with X minus. We can also have the iodotriazole, and here see that this can function as a halogen bond donor, which can interact with an anion, and we can now activate this further by taking the methyl substituted iodotriazolium and this can also, so this is the iodotriazolium. So, the iodotriazolium compound can now also function as a halogen bond donor. This is even more activated and then can accept anions. So, we have looked at the vast spectrum of these particular anion hosts which are now present in the systems, and keeping this in mind, we would now like to appreciate further the application of anion sensing and cation sensing in biological systems. Now, we would also now like to look at biological anion receptors. So, here what happens is that we know that in case of a biological system, we have got an enzyme, and the enzyme is the host, and now we can have different kind of anions which will come and bind to this particular host. Now, it is very important that in case of protein structures, the structure need not be a very rigid structure. It can be a flexible structure which can undergo conformational change, so that it can accommodate the anion binding. So, the prerequisite of pre-organization, which was very important in case of cation binding host, is not a very strict requirement here. And many times in protein structures, it is the change, the conformational change in the tertiary structure of the protein, which is very important to accommodate the anion binding. Another very important thing in biological receptors is that they play a very important role in enzymatic catalysis. So, here you see that if an anion has been captured, so there is complexation, and then there is this anion has to be transported to a particular place, and then it has to be released at the site of action. So, this process of complexation, transport and decomplexation plays a very important role. So, say the anions are initially solvated, you have the desolvation, the anion is captured by the biological protein, or which we call as the enzyme, then it is transported to the site of action, and there it is then has to undergo decomplexation. So, kinetics play a very important role, kinetic selectivity plays a very important role. Thermodynamics is also important to efficiently bind the anion, but then this binding should not be very strong, otherwise the anion will not be released at the site of action. So, this kinetic selectivity is very important. So, proteins in this case need to be more like thread like flexible structures which can have conformational change, so as to accommodate the anions. And when an anion binds with a protein, it is primarily dictated by enthalpic considerations, where a large number of hydrogen bonds and ionic interactions are present which stabilize the anion at the point of binding with the protein. And also we would like to keep in mind that a protein structure is a case where you have the primary structure, the secondary structure, the tertiary structure and the quaternary structure, and it is a very specific mode of self assembly of the protein. This word self assembly is something we will explore more in the next week, and how it is different from molecular recognition. In case of self assembly, each step is very precise, and the information coded or contained in the system is also very specific, such that if first the primary structure is not formed, then the secondary structure will not happen, the tertiary and the quaternary states will not be reached. So, the primary structure is essentially the sequence of amino acids which will form the peptide bond, and depending upon how many such amino acids are formed, a large number of such amino acids will give you the primary sequence of the protein, which effectively can be considered to be a long thread like arrangement of amino acids to give you the primary structure. Now, this chain of amino acids or these long chain peptides which give you the protein, they can actually they can now start getting coiled up. They can now start getting coiled up in different ways, and this can give rise to the formation of beta helices and alpha sheets. So, the formation of these alpha sheets and beta helices is what gives the primary, we can say essentially the primary or the building blocks, which gives rise to the secondary structure, that means the formation of these beta helices and alpha sheets. And the manner in which these alpha sheets and beta helices further fold the conformation of the protein to give it to take it into a particular conformation, and in most of the cases it is the globular conformation or the globular shape of the proteins which is very common, and this compact conformation or the globular shape or the globularity of the protein essentially arises from the different types of folds which is undertaken by these alpha sheets and beta helices to give you the tertiary structure. And it is here at this time when you have the formation of the alpha sheets and the beta helices that intramolecular hydrogen bonds and then dispersive interactions become important. And once you have a particular protein, how it interacts with the other protein via again these hydrogen bonds, but now they are intermolecular hydrogen bonds. So, how one protein interacts with another protein, for example, in a crystal structure you have one protein interacting with another protein, that gives rise to the quaternary structure of the protein. So, in this regard proteins are very important processes of formation, very important molecules, and an extensive study of anion binding proteins has been observed, and in this regard sulphate binding proteins have been recognized. We call it as SBP, and here you can see that the active site is P double bond O, OH, O minus, O minus, where you can see that this functions as an acceptor of hydrogen bonds. So, it can accept hydrogen bonds from say water molecules which are present in the protein structure. So, protein structure has got large number of water molecules. So, these anions will get extensively solvated by the water molecules, and in turn these hydrogen bonds, the OH, these are the donors, they can now interact with the acceptors on the protein site at a different protein sites. Similarly, this is an acceptor. It can now interact with different donors via hydrogen bonds. So, we can have this H PO4 minus, we can also have P double bond O, OH, OH, O minus. So, we can also have H2 PO4 minus, we can have H PO4 2 minus. So, these kind of species are present, and what has been observed in the case of sulphate binding proteins, there is a specific case where it has been showed that in a particular protein at the active site the phosphate is surrounded, this phosphate is surrounded by 12 hydrogen bonding interactions, out of which, and these hydrogen bonding interactions are such that the N/O dot dot O. So, you can have NHO or you can have OHO hydrogen bonds. These are between 2.6 to 2.9 angstrom, and we have 7 such hydrogen bonds which involve the arginine side chain or the protein main chain. So, the protein main chain is the CONH sequence, and the arginine is one of the amino acids which has a NH donor which can interact with the phosphate unit, and 7 such hydrogen bonds are there. There are 4 such hydrogen bonds where 2 are coming from the serine OH and 2 are coming from the threonine OH, and 1 which is coming from the oxygen atom from the carboxylate ion, and this kind of very specific hydrogen bonding pattern between an anion with an active site of the protein gives the protein its selectivity, and the fact that proteins function in a certain way is because of these distinct hydrogen bonding arrangements which are of extreme relevance. So, this is with respect to sulphate binding proteins. There are another important class of proteins which are called phosphate binding proteins. In case of phosphate binding proteins, we have got sorry, I made a mistake here. We have got, so okay, we discussed about, so we will, I will consider this as phosphate binding protein. Now, we will go to the sulphate binding protein. So, in the case of sulphate binding protein, we have now the sulphate anion, and this can now interact at the active site with different kind of amino acids, and again you can have different kind of hydrogen bonds which are present here, and there are 7 such hydrogen bonds which are formed these come from the backbone NH, serine OH and tryptophan NH. These are all the hydrogen bond donors. And this is what gives this particular protein its selectivity. And what has been observed is that this function is very specific to this atomic arrangement, because if you replace the OH of serine with SH, or you replace it with CH3, or you replace it with glycine, in this case it is alanine. Okay. So, when you do this simple replacement of one of the atoms, say OH with SH, SCH3 or glycine, then it reduces the affinity for sulphate binding. So, that reduces the affinity of the protein for sulphate binding, and here it is drops by a factor of 3200, here 100 and here 15. So, this is also very important. It is a slight modification of the electronic environment. We replace this OH with sulfur, SH dramatically or drastically reduces the affinity of the same protein for the sulphate, because you have now modified the donor. When you have replaced oxygen with sulfur, it now becomes a much weaker donor compared to OH, which is a much stronger donor, and also it forms much stronger hydrogen bonds compared to that of SH. So, this plays a very important role in the anion recognition at the sulphate in case of sulphate binding proteins. So, what we have learned now is that in case of proteins particularly, we do not need pre-organization, and we see that essentially these are stabilized by the formation of large number of hydrogen bonds. Enthalpy considerations are very important, and more is the extensive hydrogen bonding of hydrogen bonds, and even we have got van der Waals interactions between the hydrophobic parts of the protein. Then these will tend to be formed and favored, and that will actually stabilize the aggregation. Now, the next thing we would like to further discuss here is we would like to take up the topic of transmembrane anion transport or cation transport. Now, this is again very important, because we have now looked at anions as a whole, we have looked at cations as a whole, and this anion transport and cation transport is very important in biological cells. And in a biological cell, what essentially happens is that we have got an aqueous interior which is separated from the exterior by phospholipid bilayer cell membrane, which is around 5 to 6 nanometer in width. So, outside the cell it is an aqueous environment, and inside is an aqueous environment, and in between we have got this phospholipid bilayer cell membrane, and it is through the cell membrane that the communication takes place. Different kind of active ingredients have to come from outside to inside, or they have to go from inside to the outside via this bilayers. So, the formation of this bilayers is extremely important, and the phospholipid bilayer is chemically containing a phosphate linkage CH2. So this is a phospholipid bilayer, and this we can represent it as a negatively charged species which represents the phosphate anion, and then we have this corresponding hydrophobic backbone. For the case of simplicity, I will also write it as follows. So now we have this bilayer arrangement where we have these this is just for the sake of simplicity, because we know that there is a hydrocarbon chain here, and we can now have okay. So we have the aqueous region here, we also have the aqueous region here, and in between we have got the bilayer, and we can actually have the transport for example of we can have the transport of a cation, say potassium, or an anion, say chloride, and it transports from say the outside to the inside of the cell. For example, this is referred to as the uniport mechanism. We can also have a way of transport such that you have the cation and you have the anion, and they both are transported to simultaneously through this particular bilayer, and this is referred to as the process of symport, because they are being transported in the same direction. On the other hand, if they are being transported in the opposite direction, say the cation moves in this direction and the anion moves in the opposite direction, that is referred to as an antiport mechanism. And overall, this symport and antiport is referred to as co-transport. So this symport and antiport mechanism, where the anions are going or moving or being transported from one side to the other, and they are being transported across this bilayer, is a very important process in biology and in maintaining the equilibrium of the system. And so we will look at how does this for example the cation move from the outside to the inside of the cell, and we will look at these mechanisms in the next lecture. Thank you.